Thank you very much. Thank you for inviting me to talk in this symposium. So yes, changing tracks now, and we've heard a little bit about fungus from Matthew at the very beginning, and of course a lot of the previous talks have really set the scene for some of the things I'm going to be talking about. I come from the Mycology Reference Centre in Manchester, which is part of what's called a National Aspergillosis Centre, a referral centre for chronic pulmonary fungal diseases in Manchester, and we're then part of the much bigger Manchester Fungal Group, about 80 people working on different aspects of fungal disease, both in the clinic and also in the laboratory. And I suppose my first contact with Sergi Honey was through Jonathan here, and we've been interacting pretty closely since then. So this is my disclosure. I'm very grateful for the samples of Sergi honey that we have been receiving. And we, being really a master's student, not myself, a master's student, we run a master's program in medical mycology. And this particular student, Zain Al-Hindi, one of our students from Saudi Arabia, she did the work, very preliminary work that I'm going to describe to you over the next few minutes. So I'm not going to be unkind and pick on those few people in the audience I know would know the answers to these questions, looking around, but nevertheless, this shows you the spectrum of fungi, and we've been familiar for a long time that Candida can form biofilms on inanimate surfaces, and of course also on host tissues. And also basically chronic aspergillosis, aspergillomas and chronic pulmonary aspergillosis is a biofilm disease. We do recognize that now, and in vitro a lot of work has been done on aspergillus biofilms. My focus is going to be on Fusarium, and of course Fusarium has really hit the headlines over the last couple of years as a global outbreak of fungal keratitis caused by contaminated contact lens sterilizing fluid. So big numbers, a devastating disease, and of course globally fungal keratitis caused by Fusarium and other fungi, about 1 million people are known to suffer from fungal keratitis. So this could long-term be a clinical situation where a product like Sergi honey may be of some use. And then of course there are other pathogens here which we don't have time to go into, but most of these, yes, do form biofilms. Quickly then, the size of the problem, just to really sort of set the scene. I'm not suggesting that all of these people could be treated with alternative products like Sergi honey, but over 300 million people are affected by a serious fungal infection, Candida aspergillus and others. And we can see that sort of broken down a little bit here. Candida, of course, candidemia being the main infection that we see here. And here's fungal keratitis, the sorts of numbers that we're talking about. And yes, people die from fungal infections. Meningitis caused by Cryptococcus, aspergillus, chronic pulmonary aspergillosis. These are the patients that get referred to us from all over the UK. And also pneumocystis, of course, quite a high mortality in AIDS patients. And Candida bloodstream infection, high mortality, even though we do have some improved diagnostic tests, but still over 100,000 deaths. And SAFs, severe asthma that is complicated by fungal sensitization. This is a very serious respiratory problem that we do see in our clinics in Manchester. So in terms of fungal biofilms, the tsunami in 2004 in Thailand and elsewhere in that region really highlighted the fact that necrotizing fasciitis as a result of being caught up in the tsunami was a major sort of problem. And also here we have a lady who survived that tsunami. This lady was treated with a long course of liposomal amphotericin B. And I think a systemic drug is needed in these sorts of situations. And then, of course, we may hear a bit more about this in a talk later on today. Of course, theatres of war have highlighted the problems of devastating soft tissue diseases caused by a variety of fungi, agents of mucormycosis primarily. So the background to our work, as I say, very preliminary data. This was really, and this has already been talked about already, how important reactive oxygen species are in host immunity. Fungal phagocytes are very effective in interacting with Candida cells, with Aspergillus cells, and very effectively ingest and kill fungal pathogens. And of course, what we're trying to do is here, we're trying to prevent, with whatever treatment we're exploring and alternative treatments, we're trying to prevent the germination of spores. So these could be conidia on a wound, on a burn, trying to prevent that whole process taking place, which in the absence of effective host cells is an unstoppable sort of process. Germination Aspergillus, for example, will grow at 2 millimeters per hour on a Petri dish, and I suspect even faster on a tissue surface or in tissue. So this is what we're really trying to do, is to stop this germinative process. Yes, so fungi form biofilms, and here is just one example. This is not a fusarium biofilm, which is going to be the focus of my talk, but this is an Aspergillus biofilm, this complex community of conidia here. We can see lots of conidia here, but also these branching mycelium. The whole structure is enmeshed in this extracellular matrix. So this is quite a formidable target, but perhaps closer to what we're really trying to demonstrate than just using, as Beryl has already told us, looking at planktonic cells in standard MIC-type testing systems. So, initially, things were looking pretty good. So this is Sergi honey and Scedosporium apiospermum, a cause of a variety of infections, pulmonary infections and systemic infections. And as was shown right at the beginning of today by Matthew, very simple well diffusion types of experiments, ending up here with this large zone of inhibition with 100% Sergi honey added to the well of this plate. So quite a remarkable zone of inhibition there. And the same with Candida. Some activity here. We have Candida tropicalis. Again, 100% honey. We've got this zone of inhibition as a result of the product diffusing out of the central well here. Disappointingly, and I think this is something we have to re-explore and spend far more time on, this is Aspergillus fumigatus, and this slide would suggest there's no activity whatsoever using the same sort of experiment with 100% honey here. This could just be because it's a very, very high inoculum and other factors which did not demonstrate any activity of Sergi honey on Aspergillus fumigatus in this particular panel. So we know that there are cutaneous fungal infections, biofilms are present, especially where there has been delayed wound healing. We know that fusarium species form biofilms within the chronic wound infection, and because of antimicrobial resistance and also because of antifungal resistance, we're looking at 20% antifungal resistance in our patient population because of long-term azole usage. Azoles are the main drugs that are used. Why? Possibly driven by the use of azoles in agriculture. Millions of tons of azole drugs are used to control plant pathogens, and we believe this drives resistance in Aspergillus fumigatus, which of course our patients inhale and are exposed to. So fusarium, for example, it produces these sporodocia on cutaneous wounds. So this is a high-resolution picture of aerial hyphae growing out from a wound. And if you look back at the literature, just one paper of many, back in 1991, a 10-year experience, fungal burn wound infection. So this really is sort of setting the scene for looking at alternative treatment modalities, fusarium and Aspergillus primarily, but also Candida to a much lesser extent, agents of mucormycosis. And of course, these were confirmed in this paper by histopathological confirmation. And the treatment options are systemic antifungals, of course, topicals like cotrimazole and topical nystatin, but also amphotericin B is used a lot for these more deep-seated infections. So the aim of the study, let's see if Sergi honey had any effect against fusarium biofilms. We chose fusarium for the reasons I've explained. Also, fusarium biofilms are very easy to establish. We've had a lot of experience now in establishing Aspergillus biofilms in vitro. So there are very, very few publications looking at fusarium biofilms, but these were set up in the same sort of way. Then the question is, how do you assess the effect of whatever you're trying to look at in terms of its antifungal activity? There are various ways, of course. You can disrupt the biofilm, and you can look for the viability or lack of in your biofilm, or you can use metabolic assays like the XTT. I'm not going to, because I can't remember, I'm not going to give you the full name of that assay. It's a couple of lines long, but it's known as the XTT. It's an assay to measure metabolism or the lack of. Then, of course, you can look at the way in which, in our experiments, Sergi honey was disrupting the cell wall, and galactomannan is a major building block component of fungal cell walls. So is glucan. This is a pan-fungal diagnostic test that we use in routine diagnostics, but also is a useful biomarker for cell wall disruption. And then, of course, we can also compare the biofilm with the planktonic cells. So here we have, first of all, again, back to the simple experiment. Here we have 100% honey against Fusarium oxysporum in this particular case. This seems to be the major fungal pathogen that's involved in various wound infections and also in these keratitis cases, showing quite nicely how active Sergi honey is against Fusarium. So this was the methodology. Oxysporum and Fusarium solani, the sort of concentrations of cells that we use for forming the biofilms, equally the work that was done with planktonic cells, and the concentrations, very simply liquefying the Sergi honey and using these various percentage concentrations, compared with amphotericin B, and also compared with pimarosin nicomycin, which is the standard treatment used for fungal keratitis caused by Fusarium and other molds. And then assessing either with standard UCAST designed UCAST standards, or using these biomarker assays. So very briefly then, it's not a typical dose response curve. 100% honey did not seem to reduce the amount of growth and metabolic activity, surprisingly, but 50% concentration of the honey there was quite a marked difference between this and the controls. For reasons we don't understand, why didn't the 100% concentration of honey completely shut down the metabolism of these biofilms? And the same sort of result is here. We see most activity on the cell wall with 50% honey, as shown here with this galactomannan index. The more galactomannan that's released, the greater the destruction of the fungal cell wall. And here we have Fusarium solani. Here we have the same sort of effect here. The 50% honey seemed to be the most active, and that was also shown here in terms of the galactomannan. And this is Fusarium oxysporum, an older biofilm, 48 hours treatment. So this is really treating the biofilm, well established with the Sergi honey. And we can see here that this concentration of honey, 25%, seemed to be the most active. And then finally, looking at the XTT assay with Fusarium solani in this sort of way, again, the lower concentration of the honey seemed to be the most active, and inversely was the most efficacious in disrupting the galactomannan from within the cell wall of the organism. In terms of MICs, we can see here sort of reasonable results of the honey compared with amphotericin B and nicomycin, and also the release of beta-1,3-D glucan from the biofilms. The honey seemed to release the glucan, and that was shown by these very high levels of glucan released from the biofilm surface. And then looking at the microscope here, we can see, down the microscope, we can see here the positive control here. We can see again the effect of Sergi honey on these biofilms. So to finish off then, there's a suggestion from this work, as I say, very preliminary, that Sergi honey seems to have an effect on Fusarium solani, both planktonic cells and oxysporum, and also in the biofilm state of growth. and so it seems to have both anti-biofilm and antifungal properties. Galactomannan release was demonstrated, and this possibly may be an erroneous result because galactomannan is a very common molecule, and it could be present actually in the surgery honey itself. So that's something else we really have to look at if we're going to carry on using galactomannan release as a mark of cell wall disruption. And also glucan was released at a higher level. So as a result of all of this, all universities, of course, are keen on press releases, and Manchester is no exception. They heard about this little project, and this was written into a press release here. This came out on the 5th of February and was picked up by over 50 different science journals around the world. So I think this has been quite good exposure for the work that we're trying to achieve in Manchester, and of course good exposure for the product itself. And if you're interested, if I've sort of given you an idea of what mycology is all about, or you or your junior staff are interested in spending a bit more time studying biology, this is the structure of our one-year masters, taught masters in Manchester. Thank you very much.